Good morning, all. Let's go ahead and get started. We are rapidly approaching the end. Today is Monday, May 3rd, 2021. We are on to our second lecture of the reproductive system, and then we will finish off the boy version and move on to the woman version of that. We have a couple more assignments left, 2 more labsters. Again, remember, make sure you do all the activity and get it 80% correct. If you don't get it 80% correct, remember you can do it again to make it 80% correct to get full credit. And 1 more unit review just for the heck of it. Then we have nothing but exams. Wednesday the 12th, lab and lecture exam. You know what to expect from that. And then Wednesday the 19th, you have your cumulative final exam. 100 multiple choice questions, 100 minutes to answer them, and again, covering everything we have covered for this class. All right, questions on any of that? All righty, excellent. Then let's pick up where we left off.
Actually, I did have a question.
Yes, go ahead.
For the Friday labs, how do we let you know that we attended?
You don't.
Okay,
As long
you
as
don't have
you
to.
are telling, as long as you are checking in with Jeff when you get there, and then checking in with Jeff when you leave, he has been keeping track of your hours. And so he will then, at the end of the semester, he emails me everybody's numbers, and then I input that into the computer. Also, if you've done the Science Success Center, and the Science Success Center has contacted me, they send me an email once you've completed that. Again, I purposely, well, first, I haven't gotten the information from Jeff yet. He only sent it to me at the end of the semester. But also, if you've completed the science skills, I purposely don't put those points into the grade book because I don't want you counting on them. I want you getting the grade that you want in this class without them. But if you fall a little bit short, then you have things like the science skills, you have things like the open lab to help you to be successful with them. So yeah, so you don't have to worry about that. As long as you're checking in with Jeff, then he will pass that information on to me. Great question. Any others? All righty. So last time we were working our way through the male reproductive system. We had talked about mitosis, meiosis, the process of spermatogenesis, the accessory structures. Pretty much the only thing that leaves us with an anatomical standpoint for the male is, of course, to talk about the penis. Of course, as we all know, the average penis is 2.5 feet long, the width of a soup can, prehensile, jointed twice in the middle. Right, they're true beasts. And of course, its function is twofold. Obviously, it serves a function in the urinary system for the release of urine, but it is the copulatory organ for the reproductive system, whose job it is to present the ejaculate, the semen, to the vaginal canal for fertilization. The penis has basically 3 distinct regions to it. It has the shaft or body of it. It has an enlarged head region known as the glans. Which is covered, although this illustration doesn't show it. The next one I believe that we have does. There is a fold of skin that extends around and almost completely encloses the glands of the penis, what is known as the prepuce, or what is it commonly referred to as also?
Foreskin.
Yeah, the foreskin, absolutely. For medical, for cultural, for religious reasons, that is often removed in a process known as
circumcision.
Circumcision, there you go, exactly. And then obviously there is a root to it as well. Now, these are the external structures. However, if we were to look internal to it, we will see that there are 3 areas of spongy erectile tissue. Here we see that kind of longitudinal view we were seeing before, where we can see some of the anatomy of this. And notice this one does show that a flap of skin that extends around the glands of the penis. That is that prepuce. And this shows our 3 distinct regions that are of that spongy erectile tissue. This is a loose tissue that, like a sponge, can absorb fluid. In this case, that fluid is blood, causing it to engorge and enlarge. Here, we see it in a cross section of the penis, so we can see more of the relationship of these erectile tissue regions to each other. For starters, we have a single region known as the corpus spongiosum. Notice this corpus spongiosum surrounds the urethra. Why would we want erectile tissue to surround the urethra?
Keep it patent.
Yeah, exactly, right? Copulation, when done properly, should be vigorous, right? And since the point is to present the ejaculate to the female reproductive tract, if during the process of copulation, the urethra is collapsed, then the ejaculate cannot be released. And so to maintain the integrity and keep that urethra open, allowing for that release, we have this erectile tissue that surrounds it and maintains the integrity of it. It is also the corpus spongiosum that enlarges and expands to form the glands at the distal end. Notice also it enlarges proximally as well, and that proximal enlargement is what is known as the bulb. And the bulb is one of the major structures that attaches it to the pubic symphysis, as you can see its relation here to the urogenital diaphragm. Superior and lateral to the corpus spongiosum are the plural corpora cavernosa. Of course, the singular would be corpus cavernosum, but these are two of them. So the plural corpora cavernosa, or what are also known as the dorsal bodies. Their primary function is to give the penis the rigidity necessary to act as a copulatory organ. Hopefully, since the last class, you've had the opportunity to examine the raphe of a male scrotum, to observe the contraction of the cremaster muscle, to palpate the ductus deferens and all of those things. And while that occurred, the penis associated with all of those structures may have become engorged. And if you palpate the penis, you can actually feel these three distinct erectile structures in it. So while obviously it looks like a cylinder, when you palpate it, you can actually feel these distinct regions within it. Again, their job is to turn the penis into an erectile structure, but also, as you can see proximally, the taper off to the end, forming these two tail-like structures known as the crura, singular being crus, plural being crura. Great question. Absolutely. I don't want videos or anything like that. You're welcome to keep those for yourself. I'll take your word on it. The crura and the bulb collectively form the structure that we call the root. And so again, they attach and anchor the penis to the levator ani muscle and to the pelvic bone, to the pubic arch. So collectively, they form what we call the root of the penis. So those three structures come together to form that. All right. Now, as you can see, actually, we'll take a look at this histologically, I think is one of the nice ways to do it. As I mentioned, the importance of this tissue is it is erectile tissue, meaning that when it becomes engorged with blood, it enlarges. However, we call it spongy tissue because it acts like a sponge. And if anybody's ever washed dishes before, when you take that sponge and put it in the water, what happens to it?
Expands.
Yeah, it expands. Absolutely. Excellent. Does it become rigid as a result of that when it expands? No, it's still floppy all over the place, and that wouldn't make a very good copulatory organ. So what you can kind of see from the illustration of these dorsal bodies, and we'll see this when we take a look at it histologically, you will see that there is a collagenous frame to this tissue. So that what happens is, as it expands, it pushes against this fibrous collagenous structure. And as it forces against that, that is what gives it its rigidity. So if we notice, and I think now is probably as good a time as any to do this, let's go ahead and find our penis. Here we go. Here we see a cross section through the penis. Here we can see, so this here is our corpus cavernosum. Here is our second corpus cavernosum. Here is the urethra, and around the urethra, we can see the corpus spongiosum. So we see that erectile tissue, but as you notice, what you can see surrounding it is this lightish colored, whitish color. It even actually has a septum that separates down the middle. It does look a little surprised, like a face. Absolutely. Mostly because males are surprised when females say yes. Is that you have this fibrous collagenous structure known as the tunica albuginea. Right? Tunica, again, tunic outer layer, albuginea because it is whitish in color because of the collagen. And so this forms the structure, the frame that then as that erectile tissue expands against it, it forms the rigidity, makes it turgid so that it can be the copulatory organ that it needs to be. I think I have one other picture here that does a nice job of showing this as well. Again, here, different stains, slightly different coloration to it. So we can again see, not quite as distinct, but we can see that tunica albuginea around the center. But the other thing that I wanted to show you is if you look closely deep in this dorsal body, in the tunica, pardon me, in the corpus cavernosum is a large blood vessel, and that blood vessel is the blood vessel that is going to release the blood into this tissue and allow it to expand. And what might we call a blood vessel like that?
An afferent artery?
Not a bad guess, but I was thinking more in terms if you had a list of anatomical terminology in front of you, you might be able to figure out what that particular blood vessel that allows the blood into the corpus spongiosum might be. In the corpus cavernosum, I should say.
The central artery?
Yeah, exactly. That central artery, exactly. That is going to be able to release all of that. So you can typically see, and again, it depends on the plane of section. I don't see a good view of it here in this particular one, but this is a really nice image of it for that one there. So that is something that can be useful as well. All right, excellent. So we've taken a look at that. We've seen the anatomy of this. Let's talk a little bit about the function of it. Like I said, its job is to be the copulatory organ. And if you remember way back in 430, when we talked about the autonomic nervous system, I promised you we would use the mnemonic point and shoot, right? Because when we talk about arousal and ejaculation and the control of that process, it is important to remember point and shoot, because that's not how you spell point. Point, the arousal, the erection, is a parasympathetic reflex, whereas the ejaculation, the shoot, is a sympathetic reflex. Arousal, erection, or a process called tumescence, like I said, is a parasympathetic reflex. Basically, upon arousal, a couple different things occur. The first thing that happens is that our parasympathetic nervous system releases nitric oxide, which causes vasodilation to those central arteries. Now, if you remember of the erectile tissue, oops. This is a somewhat unique characteristic because as you remember when we talked about in 430, most blood vessels are controlled autonomically only by the sympathetic nervous system. So if you remember, most of the blood vessels, the redirection of blood during that fight and flight response is solely controlled by the sympathetic nervous system. But as we know, most means not all. And so as we talked about in 430, and I'm reminding you about here, the erectile tissue, and again, in males, obviously that means in the penis, but also in the females, in the clitoris and the labia, that erectile tissue that becomes engorged upon arousal is actually a parasympathetic reflex. And that causes those blood vessels to dilate. And as those blood vessels dilate, those areas become engorged with blood. Again, being engorged with blood makes it larger, but that doesn't necessarily give it the rigidity. So remember, as we also talked about, as it engorges, it is going to force itself against that collagenous structure, that tunica albuginea, and that is what gives it its rigidity. However, it is important to remember that, well, actually, we can say this too. Not only does, as it engorges and enlarges, it gains that rigidity by pushing against that fibrous structure, it also causes the erectile tissue to basically compress the drainage veins. Of course, that means we get congestion in the area, which normally we think of as a bad thing, but in this case, it keeps the blood in the region to help to maintain the rigidity of the penis. All right, again, as we've talked about many times in this class and in 430, the more we know about the body, the more we try to manipulate it to our advantage. And there is, and I'm always hesitant to use the term toy when they talk about this, an instrument that sometimes is used to take advantage of that, right? A ring-shaped structure that can be put at the base of the penis to increase the compression of those drainage veins, allowing the male to stay more rigid for a longer, prolonged period of time, right? And that all sounds fine and dandy, but remember what we are doing here is congesting the area with blood. And the problem with that is, as we know, blood is necessary for regions to provide oxygen, to provide nutrients. And while it's all fine and dandy to try to maintain that, to be able to prolong the enjoyment of your partner while this is taking place, these things are not one size fit all. And there have been many instances of individuals who have used this type of device, and that congestion becomes chronic. Becomes an issue where after copulation occurs, it still is unable to drain, right? And if it stays congested with blood for a prolonged period of time, lactic acid can build up, the tissue can be damaged as a result of that. And so how do they typically have to resolve that? Yeah, they have to drain it. So you go to the doctors, exactly, more than 4 hours, you go to the doctors. They use a very large bore needle, which they will insert into the erectile tissue of the penis to extract some of that blood, to decrease the size of it enough so that that ring can then be removed. But by the time you've gone to the doctor, you've probably caused some damage. The insertion of the needle can cause some damage. And of course, this is highly specialized tissue that doesn't regenerate. Instead, it heals via fibrosis. So if you damage this tissue, you're going to replace it with scar tissue. And is scar tissue going to function the same way as the tissue that was there before? No. And some of the damage that can be caused by that is permanent, right? Hardly the type of thing that I would call a toy. All right. Just do your Kegel exercises and you'll be fine with that. All right. Now, again, the goal of this is to maintain the rigidity, but let's not forget that there is also some glandular activity that is going to take place as well. In particular, during the arousal, that comes in the form of the bulbourethral glands. Remember, they become active on arousal and are responsible for that pre-ejaculate. Remember, as we talked about, it is a very viscous, mucousy, alkaline secretion. And as we talked about, its job is to cleanse the urethra, remove the acidity of the urine from it, and to act as a lubricant for the copulation to take place. So arousal, again, involves both dilation of blood vessels, redirection of blood, and also glandular activity. And again, you may not have thought of this in these terms, but I know you were aware of this because, like me, right, just this past week, because I've gotten my vaccine, I got to take that special boy out to a special and all that all important third date. And when you take them to that third date, you always take them to a really nice restaurant. And of course, when you take that boy to the third date, he knows you're expecting something in return for that, right? And so he may be a little nervous. And if he's nervous and his sympathetic nervous system is a little too active, as we know, there's that autonomic tone. So if he's a little too nervous, he may have trouble with that parasympathetic activation, and he may have some difficulty rising to the occasion, right? So what do? You know, you do the same thing I do. You ply him with alcohol. Then he's going to be a little bit more relaxed, right? Don't go too far, because then you have that whole whiskey thing to worry about. But ply him with just enough alcohol to take the edge off, and then you should be able to get what you need out of him. All right. Excellent. All right. Questions on that? You saw how much Laura laughed. You can tell she does that a lot. All right. Excellent. So arousal, the point is a parasympathetic reflex, but ejaculation is a sympathetic reflex. And not only is a sympathetic reflex, but as we have talked about in the past, we think back to, again, 430, it is a positive feedback reflex, right? Remember, with a positive feedback reflex, we get a disturbance, we get some change or effect that occurs. And rather than trying to cancel that disturbance, we enhance that disturbance more and more and more. Remember the two examples that we commonly use in 430, because when you're dealing with 430 students, they're not mature enough to have these kind of conversations. They think of it in terms of blood clotting and parturition, right? Giving birth, right? But ejaculation is also a positive feedback. You get that stimulation, that arousal, that enhances further arousal until eventually, like all positive feedback reflexes, right? Some event occurs and the body's brought back into homeostasis. Of course, in this case, right, that event is the ejaculation. So arousal continues to increase and enhance in that sympathetic nervous system. And again, much like arousal, we are going to get both glandular activity and muscle activity. Now, of course, the muscle activity of arousal is the dilation of smooth muscle, bringing more blood to the area. And we see a similar thing here as well. For starters, upon ejaculation, one of the first things that is going to occur is the contraction of the smooth muscle of the internal urethral sphincter. Why would we want to contract the internal urethral sphincter?
Because that's where it separates the bladder from the urethra.
Yeah, absolutely. And so why would that be important? Why do we want to separate the bladder from the urethra?
Because you don't want it diluting what you're releasing.
Absolutely. That is definitely one of the points. You do not want urine being released at the same time you're releasing the ejaculate. So absolutely, that is a huge advantage. But it also works the other way as well. At the same time, when we're releasing the semen, when we're releasing the sperm and the glandular secretions, we don't want them going into the bladder as well. So basically, it helps to separate the bladder, the urine from the semen. And so by closing it, it makes sure that the semen can't go into the bladder and that the urine can't come into the semen. Absolutely. So it helps to separate those two. We are going to get smooth muscle contractions of both the ampullus of the vas deferens. Again, remember, these started on arousal. So again, let's remember that, again, withdrawal is not a viable method of birth control. We start to get some minor contractions of those ampullas upon arousal, but they become more powerful upon ejaculation, releasing the sperm into the urethra. We also get the contraction and the release of the fluids from the prostate and from the seminal vesicles. or the seminal glands. So we get the release of that as well. And we get the movement of these fluids through the ducts of the urethra and out the penis via peristalsis, right? So again, it's not like turning on a hose where you get a continuous flow. It is a, right, pulsatile nature to the expulsion of that ejaculate via the peristalsis of the penis. So we get all the smooth muscle activity, all of this glandular activity causing the release of the ejaculate. And then what happens?
Then you have a smoke?
There you go. Or you go to sleep or something else. Absolutely. After ejaculation for a male, the male enters a state of detumescence. Detumescence, again, is caused by the sympathetic nervous system. During this period of time, the arousal is lost. The erection is lost as a result. And again, this is primarily due to, again, smooth muscle contractions. For starters, the blood vessels feeding into the erectile tissue constrict. That decreases blood flow to the area, so less blood goes to the area. At the same time, a smooth muscle within the erectile tissue itself constricts, draining the area and basically forcing those venous drains open.
Is that what the dartos muscle is in the...
No, remember the dartos muscle is in the scrotum.
Right.
So this is within that tunica albuginea, within the tunica and around the tunica albuginea, within the erectile tissue itself. There is some smooth muscle that constricts, that squeezes that sponge, forcing the blood out of it and forcing open those drainage veins so that the erection is lost.
In the picture you showed a few slides ago, have it labeled as as a dartos muscle?
In the histology one?
No, it was a cross section, like a textbook picture, showing the corpus cavernosa and the corpus spongiosa, and it was labeling the different that one right there. Yeah, see, it says dartos muscle.
No, it does say dartos muscle. Interesting. So yeah, apparently the muscle that surrounds the penis is, but no, in this case, and I guess that would play a role in it, but also within the even though this picture doesn't show it, within this erectile tissue itself, there is also smooth muscle in here. So the smooth muscle in here would also, this, I guess, based on the name, would also be smooth muscle helping to constrict it, but also within the erectile tissue itself, there is smooth muscle that helps to squeeze that sponge and again drain the blood out of it.
Okay, so the muscle that you're talking about is in the
erectile tissue itself, in the corpus.
Yeah,
exactly, and the corpus cavernosum and all of the erectile tissue. Exactly. Excellent. And once the erection is lost, is it able to be reestablished? How long does this detumescence last?
Give them a few minutes.
A few minutes? Well, I guess it depends a lot on the individual, right? For some of them, it may be as short as a few minutes. Age, certainly things like that can wait, be a matter, right? Till the end of Game of Thrones often is enough time sufficient to be able to reestablish. But regardless, there is essentially, and again, this obviously isn't the technical term of it, but we can think of it in this terms because we understand it, basically a refractory period. Right? If you think back to when we talked about action potentials, or we talked about muscle, we talked about nervous system, we know that there is a period of time where it loses its excitability, its ability to be stimulated and respond to that stimulus, that refractory period. Now, again, in this case, the refractory period is due to the increased sympathetic activation. So obviously, again, we have this autonomic tone where sympathetic and parasympathetic are antagonistic to each other. So for arousal to occur again, we have to suppress that sympathetic. We have to reestablish the parasympathetic to be able to, some machine guns, some of the muskets. Exactly. So again, it has to do primarily with those changes in the autonomic nervous system. All right. At which point you then nap. All right, questions on that? All right, excellent. So notice again, we are talking about changes in smooth muscle activity, changes in glandular activity brought on by the sympathetic and the parasympathetic nervous system. And not surprisingly, similar events are going to occur in the female as well. So we'll see what's similar. We'll see what's different when we get to the female process as well. All right. Questions on this? All right. Nothing I enjoy more than talking about erections and ejaculations, mostly staring at black screens. It's bad enough in the classroom when you're all staring at me, but when I got a bunch of black screens. So thank you to those of you that are on camera. Excellent. All right. So from there, the last thing that I want to talk about is we can't forget with the males. Excellent. There you go. Well, maybe he's learning something. This could be good for him. Excellent. I just wait till we get to the females and the pictures involved with that. There you go. You won't have to give them a tip. All right. So as we know, we need to talk about, exactly. We need to talk about our androgen production, primarily testosterone. What are the cells that are responsible for producing the androgens? In the test.
Are those the Leydig interstitial cells of Leydig?
Yeah, interstitial cells of Leydig, absolutely. So we have those interstitial cells. And again, of Leydig. You don't have to say Leydig, but it's fun. Excellent. And what regulates the production of our androgens again?
FSH and LH from the anterior pituitary.
Do both of those actually regulate androgen production, or just
Probably.
Which one?
Luteinizing hormone.
Yeah, luteinizing hormone. So luteinizing hormone is the one that specifically targets the interstitial cells to produce our androgens. Oops, there we go. Apparently I have that there. Again, remember, androgens are responsible for sperm maturation, not sperm production. Remember, sperm production is controlled by a follicle stimulating hormone. But androgens serve other functions as well. They help to maintain the reproductive organs, play a huge role in metabolism and growth, especially in bone, muscle, and cartilage. Right, so we get on puberty with an increase of androgens, increase in bone and muscle mass, cartilage thickens, leading to the enlargement of the Adam's apple. Remember, we also talked about how it thickens the vocal folds, so it has lots of effects on that, as well as providing those secondary sexual characteristics, production of hair, and all those other things that we talked about, the deepening of the voice, as well as sex drive and aggression. Right, all of those are things that are regulated by androgen levels. Now, I know we talk about hormone cycles a lot more in females, but males actually have a hormone cycle as well. However, for males, there's three components to that reproductive cycle. That hormonal cycle. First is that it is typically daily, whereas what is the hormone cycle for a female typically?
28 days.
Yeah, about 28 days, absolutely, about a month, right? So whereas males are daily. Secondly, males' hormone levels fluctuate much more minorly. There's very minor fluctuations in them. Typically, they're a little higher at the beginning of the day, a little lower at the end of the day, but the fluctuations are incredibly minimal. Are female fluctuations in hormones minimal?
No.
No, they're dramatic during the course of those 28 days, dramatic changes in that. And then the third major way that they are different is when does this hormonal regulation begin in males?
Puberty?
Puberty. And when does it end?
Death?
Death.
Yeah, pretty much 15 minutes after they're dead, right? Absolutely. There may be a minor decrease in overall hormone levels during the course of a male's life as he moves from 18 to 35 to 90, right? But it doesn't have to. And the drops typically are relatively minor. Same thing in females. Females start at puberty and then just keep on till 15 minutes after they're dead. No, what happens to females?
Menopause?
Menopause. And what happens at menopause?
They
No
become
longer.
superheroes.
Not so much that they become superheroes, but they have a lot more time on their hands because from a biological standpoint, they become useless, right? They're no longer capable of producing offspring. Just mean from a biological standpoint, right? No longer capable of producing offspring, right? Which gives them plenty of time, like Daniel pointed out, to become superheroes or do other important things because they don't have to deal with kids anymore. All right, so some major differences there, but let's take a look at this process in males. So actually, let's do this. Yes, Ash, you had a question.
Isn't there like when something's wrong with like the pituitary gland or something, it's like a disorder where you like either don't hit puberty or you hit it super late? Like I know it's rare, but I've seen like I think 1 or 2 cases of it.
So you are absolutely correct. Changes in either damage to or in some cases tumors or other issues in the pituitary gland can play a huge role in either an early onset, a delayed onset, or changes during the course. Absolutely. So those types of problems absolutely can occur. But again, we're going to try to focus primarily on the normal function of it. But yes, you're absolutely correct. Because as we know, it is here both in the hypothalamus and the pituitary gland where this process is controlled. So let's actually talk about that. Let's draw this. I like this picture from your textbook, but let's go ahead and draw this because you know how much I love our drawings and because we're running out of time to be able to do these things. So let's draw some boxes. You all in the boxes. There you go. Exactly. No, but we will label them because labeling is important. All right. Obviously, this is the hypothalamus. As you can tell, I spent a tremendous amount of time drawing it. Excellent. And someone remind me again when we're talking about, actually, I guess we should label this over here on the side so that we remember what we're talking about. This is regulation of
Homeostasis.
well, yeah, that works too. I was going to say reproduction function in males. So our hypothalamus produces hormones. What hormones is our hypothalamus going to produce and release? That are going to play a role in regulating the reproductive function in males.
The gonadotropic one?
True, absolutely. But remember, it doesn't produce the gonadotropins, but it does produce the gonadotropin releasing hormones, which it releases to the pituitary. Wow, that's way too big. Which it releases to the pituitary gland. And of course, the pituitary gland, anterior pituitary to be specific, is going to produce and release our gonadotropins. And of course, as you know, how many gonadotropins are there? Primarily? 2. What are they?
Follicle stimulating hormone.
Excellent. One of them is indeed follicle stimulating hormone. Excellent. And what's the other one?
Luteinizing?
Nothing.
Luteinizing hormone. There you go. Excellent. Let's start easy. What is the target of our luteinizing hormone?
Interstitial cells.
Excellent. The interstitial cells. And what do those interstitial cells do?
They release testosterone.
Yeah, exactly. They produce and release our androgens. But you're right. The primary androgen is testosterone. Excellent. And that does all of the things that we've talked about. Now, if I remember correctly, when we talked about the endocrine system, what did we say was the primary way that the levels of all hormones were regulated in the body?
Negative feedback?
Excellent. This is regulated via a negative feedback system, because while androgens are definitely a good thing, too many androgens is definitely a bad thing. So basically what happens is in this case, it is going to be primarily a humoral regulation. And someone remind me again what humoral regulation is?
When it counts the stuff in blood.
Yeah. It isn't so much that the testosterone targets the hypothalamus, but the hypothalamus counts how much androgens are present in the blood. And so if there's too much, it decreases the amount of gonadotropin releasing hormones it produces. It decreases the amount of gonadotropins that our anterior pituitary produces. Less luteinizing hormone would be produced, and our interstitial cells would produce less androgens. The opposite is true as well. If there's not enough testosterone, then our hypothalamus counts it, sees there's not enough, releases more gonadotropin releasing hormones. Our anterior pituitary releases more gonadotropins, and more luteinizing hormone is produced, and more androgens are produced as a result. So we have that very simple humoral negative feedback system for our luteinizing hormone. Very simple, very straightforward. Not a concept we're unfamiliar with at all. But that being said, questions on that side of our graph here? All right, excellent. So this side's a little bit trickier, but let's talk about what's going on here. What is the target of our follicle stimulating hormone?
The sperm or oocytes?
True. So again, ultimately, as we know, our follicle stimulating hormone is going to lead to sperm production, and it's going to regulate sperm production. But does it do that by communicating directly to the spermatogonium and the growing spermatocytes? Well, I'm asking the question, so what's the obvious answer? The obvious answer is no. So what cell does actually tell the spermatogonium and the spermatocytes what to do? The nurse cells. Excellent. Our follicle stimulating hormone, actually those nurse cells or Sertoli cells or whatever you want to call them, sustentacular cells, it stimulates the nurse cells. And you are absolutely correct. The nurse cells are then responsible for both the rate of division of the spermatogonium They're also responsible for, so let's do that, a little dash in front of it. They're also responsible for the rate of maturation, or let's be more specific, rate of meiosis. So again, the maturation of the spermatogonium, and even they control the rate of spermiogenesis. So basically, in other words, the entire process they control sperm production. But the nurse cells do one other important thing as well. While they're releasing chemical signals and the nutrients and phagocytosine and doing all those things associated with the sperm, follicle stimulating hormone also triggers the nurse cell to produce inhibin. And inhibin provides hormonal regulation of both the anterior pituitary and the hypothalamus. So in this case, it's not that it's counting how much of this inhibin is here, but this inhibin actually targets both the anterior pituitary, and as the name would indicate, it inhibits it, decreasing the rate, but it also influences the hypothalamus as well. Because again, making sperm is vitally important. And so let's think about how this would work to make sure we understand. Let's do it in a light colored and do it small. If we are not making sperm fast enough, or let's say enough, this is likely due to the fact that there is not enough follicle stimulating hormone. So not only are we not making enough sperm, but as a result of that, that also means that our nurse cells will be producing less inhibin. Of course, if they're producing less inhibin, that means that there is less inhibition of the hypothalamus and the anterior pituitary. So more of our gonadotropin releasing hormones are produced, more of our gonadotropins are going to be produced, and the levels of follicle stimulating hormone increase, which is going to bring sperm production back up. And of course, the opposite would happen as well. If we're making sperm too fast, that also means the nurse cell's producing more inhibin, which slows the hypothalamus and the anterior pituitary down. So on this side, on the left side of this, it is direct hormonal control. These are the hypothalamus and the anterior pituitary are the targets of our inhibin hormone. And that's it. Boys are just this simple. This little block diagram explains everything of how boys produce their androgens, boys produce their sperm. Like I said, we are the 1968 Volkswagen bugs of the reproductive system. Now, of course, because I drew it, I like my picture. But if we notice, if we go back to the pretty picture from your textbook, we see this exact same thing. Again, pituitary gland is constantly releasing luteinizing hormone and follicle stimulating hormone. being driven by the gonadotropin releasing hormone of the hypothalamus. And here we can see the two targets. Luteinizing hormone is stimulating the interstitial cells. Follicle stimulating hormone is stimulating the nurse cells. Ooh, we should do that too. Go back to my drawing for a second. Because remember, our nurse cells are doing one more thing as well. Not only are they producing the hormone inhibin, not only are they regulating production of, so here we'll sneak it in this way, but remember they're also producing those androgen binding proteins, which are taking, so I could put that there, not there, that are binding those androgens to bring them to the epididymis. And why do we want to take those androgens to the epididymis?
For maturation of the spermatocytes.
Right, because remember, our androgens don't help in sperm production, as we see here, but it is vital for sperm maturation. So there you go. So can't forget that. Excellent. So again, we have all of this function of the nurse cells. And so this process of androgen production and sperm production is kept relatively stable. Like I said, minor fluctuations during the course of the day, but pretty much from puberty to birth, this process continues. And here's the nice illustration, but here we have we did all of that. This is what I want. Here we go. That nice, pretty picture. Oh, there's a reversed of mine, but you see the same things. Gonadotropin releasing hormone, secretion of luteinizing hormone, which targets the interstitial cells to make testosterone and all the things testosterone does, and provides that negative feedback. And then also, as we mentioned, our follicle stimulating hormone. And again, this is the one thing that I would take exception with. They call this a negative feedback. Technically, it's not a negative feedback. This is, as we talked about, hormonal regulation, because it is not the amount of inhibin. It is that inhibin actually targets the hypothalamus and the anterior pituitary. So that would be the only thing that I would say is slightly inaccurate about this. But follicle stimulating hormone goes to the nurse cells, causing the secretion of inhibin, formation of our sperm, and production of the androgen binding proteins. All right. Questions on that? All right, excellent. Then that is everything you ever wanted to know about how boys work. So from there, we now have a basic understanding of reproductive systems. So now we get to move up to the 2022 Lexis, the female reproductive system. All right, we are going to need a fresh head. We are going to definitely need some caffeine for this. So let's go ahead. Now is a good time to take our first break. We will restart in 15 minutes, which looks like it's going to be 9:20. And at that point, I will start the recording again. So again, any other questions on the male version of this before we switch gears? The good news is there's going to be some similarities, obviously, between these two. The female version just has a few more bells and whistles, a little bit more complicated in its anatomy and its physiology. All right, excellent. Let's go ahead and take our first break, and I will see you in 15 minutes. All righty. Let us go ahead and get started. So we have finished with the male reproductive system, and we are now talking about the female. Again, like the male, there are certain similarities. The primary organs, or of course the gonads. For the females, those are the paired ovaries as opposed to the paired testes for the male. Like the male, it is also associated with a ductwork system, starting externally from the vagina with the vaginal canal into the uterus and into the uterine tubes. However, unlike the male system, as we've talked about, the uterine tubes do not directly connect to the ovaries. So as a result of this, it is an open duct system. We talked about the implications to that for females. Unlike males whose reproductive system is completely enclosed, I guess you could argue there's the side trip to the bladder, but still, for the most part, it is enclosed, whereas the females is open to the abdominal pelvic cavity. So harmful pathogens or sexually transmitted diseases aren't necessarily restricted to staying just within the reproductive system, and they can actually get into the abdominal pelvic cavity and affect the digestive system or the liver or any of the other organs that are located, the kidneys, any other organ that is located in the abdominal pelvic cavity. So that is a much more serious concern for females, and we'll see there are more protections in place in the female reproductive tract than there are in males because of this. And then, of course, also like the males, they have external genitalia, which may not be quite as prominent as the penis, but are equally important. So let's talk about all of that. Like males, we started first by talking about the testes. Let's talk about the ovaries. They are the primary female reproductive system. And of course, what are the functions of the ovaries?
Store and mature the oocytes.
Right, and produce as well. I like that. Mature, store. The developing oocytes, the immature eggs that are being produced. Anything else? Come on, I know you guys know. What did the testes do? They made the gametes, but they also did something else.
Produce hormones.
Yeah, produce hormones, absolutely. Unlike those underachieving males who only produce one class of hormones, how many classes of hormones can be produced in the ovaries?
Two.
Two, absolutely. Are estrogens, primarily estradiol, and also our progestins, primarily progesterone. So we talked about that. Excellent. Their locations are internal, inside the abdominal pelvic cavity, lateral to the uterus in their location. They need to be stabilized and held in place, but not locked in place. They need to have some flexibility of movement, not because they contract or they move or anything like that. So why do we need some flexibility of movement to the orientation of the ovaries? Is it for temperature control, the same way we move the testes around?
To allow for movement during pregnancy?
Exactly, absolutely. The females get the honor and the privilege of housing that developing baby inside of them, and that thing grows to about the size of a basketball. So obviously we needed a tremendous amount of flexibility to the organization of the organs in the abdominal pelvic cavity to accommodate that expansion. And it's held loosely in place by this collection of ligaments known collectively as the broad ligament, of course, as you know, because it's found in broads, and because it's wide and extends out. This is including the suspensory ligament, which is what anchors the ovaries in place laterally. The mesovarium, which suspends the ovaries between above the uterine tube, and the mesometrium between the ovary and the uterus itself. So again, I could try to draw this, although you know how horrible my drawing skills are. So let's instead look at the nice illustration from your textbook, that shows this. And then lastly, we cannot forget about the ovarian ligament that is going to anchor the ovaries medially to the uterus. I'll come back to the second one in a second, but let's take a look here. Here is this structure that collectively is known as the broad ligament. And as you can see, these four structures we have just identified help to stabilize the uterus in place. Laterally, you can see out here is that suspensory ligament that is going to help to anchor it and hold it in place. Notice here, basically between the ovaries and the uterine tube, which, as you can see, drapes over the top of the ovary, but is not connected to it. This over here is what we call the mesovarium, whereas inferior to that, you can see that there is this structure that kind of anchors and helps to stabilize the uterus in place next to the ovary in place next to the uterus is the mesometrium. And as I said, these two together form our broad ligament. This broad ligament is a big, huge lateral structure that actually divides the abdominal pelvic cavity along with the uterus into an anterior and a posterior region inferiorly. We'll see that in just a moment. But let's not also forget that there is a fourth structure here that is helping to stabilize the ovary in place, and that is a band-like ligament that actually directly connects the ovary to the uterus, known as the ovarian ligament. So these four ligaments all play an important role in helping to loosely stabilize that ovary in place. Now, of course, notice many of these things either directly or indirectly connect to the uterus. So it is also important to stabilize the uterus and hold it in place. And notice also, as we see from the illustration here, we are looking at a posterior view. I make a point of emphasizing this because you can see that there is one final structure I want to point out to you. And that structure here is what is known as the round ligament. And as you can see, that round ligament projects anteriorly. To help to stabilize the uterus and hold the uterus in place. So notice the round ligament doesn't directly connect to the ovary, but by stabilizing the uterus, it helps to stabilize the location of the ovary as well.
So it goes from uterus to where?
Basically to the wall of the abdominal pelvic, to the inferior lower wall of the abdominal pelvic cavity. I think, let's take a look at the next picture. Yeah, here we go. So notice here, we can see in this sagittal view a couple of things. Notice how with that broad ligament and the uterus in place, we have basically two divisions of the inferior abdominal pelvic cavity. This one back here posteriorly, which is known as the rectouterine pouch. And this one here anteriorly, which is known as the vesicouterine pouch. So we have those two indentations formed by the broad ligament. But notice also on this illustration, we can now see anteriorly how that round ligament helps to anchor the uterus towards the inferior anterior wall of the abdominal pelvic cavity. It also isn't necessarily the point that I was looking to make here, but since we're here, this is also a good, important point to emphasize how, unlike the male system, notice the bladder and the urethra are a completely separate and independent structure from the reproductive tract with the uterus and the vaginal canal. So two separate openings for two separate systems. Again, the urinary system and reproductive system are separated in the female system, whereas they are obviously connected in the male. So while the urethra in the male is a passageway for both urine and the ejaculate, the reproductive tract in the female and the urethra in the female are separate and independent. urethra in the female just contains urine. And obviously the reproductive tract is just used for reproduction. All right. Questions on that? All right. We did that. We did that. Like the testis, the ovary is not terribly impressive to look at. It's about 2 inches by 1 inch in size, and it only weighs about 1/4 ounce. But what is incredibly impressive is its function. Now, I will warn you right now, the ovary you are looking at right now is a cat ovary. We can tell that by the large number of vesicles that have actually been stimulated within it. Again, typically what they will do is they will inject these ovaries with follicle stimulating hormones in these, or the cats with follicle stimulating hormones, allowing the ovaries to mature and then harvesting them so that we get these beautiful views with all the different components to it. We'll talk about this in a minute, but one of the things that should be quite obvious when you first look at this is there is a distinct inter and external region. The internal region, of course, as we know, that deep, chewy nougat center is what is known as the medulla. And what do we call the candy-coated outer shell?
Cortex.
Cortex, excellent. So we have that. Again, just like the kidney, just like the adrenal gland, just like many of the organs we've talked about. However, and I'll erase my drawings here so that you can see it more clearly. Notice there isn't a distinct boundary between the two. There's no wall separating them. Really, the main difference are the components that are found in it. The medulla primarily just contains the large blood vessels and the nerves that are feeding into and out of the organ. Whereas as we can see in the cortex, this outer region here, the cortex contains all of the, what we call ovarian follicles. And these ovarian follicles are the developing eggs and their support cells. So 2 different regions with 2 distinctly different functions to them. And so anatomically, they look very, very different from each other as well. We may come back to this picture because it's a beautiful picture, because one of the things we'll talk about are the stages and development of our ovarian follicle and the cells inside of it. And this picture does a beautiful job of showing all of those stages. So when we get there and I'm struggling for a picture, make sure to remind me that this picture at the front was really good and I want to come back to it. Like the testis, it also is surrounded externally by 2 main layers. Like the testis, the first layer is basically the visceral peritoneum. This visceral peritoneum, basically extension of the peritoneum that wraps around it. Of course, we couldn't just call the visceral peritoneum, so it is known as the germinal epithelium, right? Kind of similar to that tunica vaginalis we saw on the testis. And like the testis, the bulk of it is a fibrous connective tissue that we call the tunica albuginea. However, unlike the testis, if you remember that tunica albuginea of the testis extended into the testis, forming individual lobules, that does not occur here in the ovary. So just the bulk fibrous outer layer, the bulk of it is composed of that tunica albuginea with basically a layer of the peritoneum on the outer surface. Oops, there we go. And again, here we see a nice, real, again, ovary. Get another cat one. Oops. As I mentioned, here the illustration does a nice job of showing, again, those two poorly defined regions, the medulla, the inner region with the blood vessels and nerves, and the cortex, which is the outer region containing those ovarian follicles, which again is a fancy term for the immature egg and its support cells. All right. And of course, the job of that is where we're going to mature the gametes. When we talk about these ovarian follicles, as I mentioned, they contain an immature egg known as an oocyte and surrounded by support cells. There are three main types of support cells that you are going to run into. The first two are actually the same type of cells, just different stages in their lives. The first are the follicle cells. These follicle cells are the immature squamous cells. They then mature into the hormone-producing granulosa cells. And if you remember from the endocrine system, the thecal cells form a layer on the outer surface and does anybody remember what the thecal cells do? You know, it seems like decades since we did the endocrine system. Anybody remember what the thecal cells did? They did something kind of interesting, so that's why I thought maybe someone might remember. The thecal cells, remember, produce androgens. The granulosa cells are responsible for taking those androgens and converting them into estrogens. So it's our granulosa cells that are responsible for producing estrogens. But they don't make those estrogens out of thin air. They actually convert the androgens made by the thecal cells into our androgens. So again, that was something we briefly mentioned, but it's okay if you don't remember that because that was a long time ago and it wasn't the emphasis there. Here, we'll talk about it and its importance much more. Now, as we can see from the pretty picture here in your textbook, there is a big elaborate development and changes that take place in that ovarian follicle that we absolutely positively have to talk about. However, before we can talk about what's going on with all of these support cells, we need to go back and talk about what's happening with the egg itself. And that is a process called oogenesis. Now, luckily for us, we have already talked about a process similar to this. And of course, that process similar to this was spermatogenesis. Someone remind me again what cell we started with spermatogenesis?
Spermatogonium.
There you go, a stem cell called a spermatogonium. Excellent. Haploid or diploid?
Diploid.
Excellent. What process did the spermatogonium undergo?
Mitosis.
Excellent. It undergoes mitosis. Let's put that there. Let's see if I can grab that. Move that there. And after it undergoes mitosis, what do we get as a result of that?
You get a primary spermatocyte and a secondary one.
Do we get a primary and a secondary as a result of it?
No, not a primary and a secondary. You get one primary spermatocyte.
Yeah, you get one cell that becomes a primary spermatocyte. And remember the other one basically, oh, thank you. The other one basically becomes another stem cell and keeps the process going. Now, of course, as we talked about, in becoming a primary spermatocyte, we undergo meiosis. So we get that cell that is got the replicated chromosomes. And our primary spermatocyte undergoes what process?
Meiosis.
Although we can be a little more specific.
Meiosis 1.
Meiosis 1, excellent. What happens as a result of this?
You get 2 haploid secondary spermatocytes.
Excellent. To essentially identical cells, although they're not identical chromosomally. I think I've made this all too big. This isn't going to fit in here. Let's see if I can cheat. Bring this up. Bring this up. Bring that up. Bring that up. Bring that up. Oops. Excellent. So we have our secondary spermatocytes. Two secondary spermatocytes. And what happens to those 2 secondary spermatocytes?
They undergo meiosis 2.
There you go. And what do we get as a result of that?
4 haploid genetically variable gametes.
Excellent. And what do we call these cells?
Gametes?
Not yet.
Unique haploid cells.
They are indeed unique haploid cells.
Daughter cells?
Say again?
Daughter cells?
True, they are daughter cells, but didn't we have a fancy name for these cell-shaped haploid cells?
Spermatids.
There you go, and more specifically, early spermatids. Because remember, there is a third process to spermatogenesis. What happens to these early spermatids?
Become late spermatids with their flagellum.
There, exactly. They undergo a change from cell-shaped cells to essentially sperm-shaped cells. And what do we call that process?
Spermatogenesis.
There you go. Spermiogenesis. Excellent. And so notice our one stem cell, or more specifically, our one primary spermatocyte has given us 4 viable gametes. All right. All of that hopefully sounds vaguely familiar. I'll pretend you all said yes emphatically. So let's talk about oogenesis. Oogenesis, in some ways, is a very similar process. We are going to start with a stem cell. And this stem cell is known as an oogonium. And not surprisingly, this oogonium is going to undergo mitosis to produce many daughter cells. And of course, as we would expect, what do you think we're going to call these many daughter cells?
Primary oogonium.
Oocytes, remember? Yeah, you're right. Primary oocytes. Excellent. So absolutely, they're going to divide. As they divide, right, they are, of course, basically body cells. So they are diploid in number, producing primary oocytes. As well as plenty more cells. So of course, they're going to divide and produce more stem cells so we can make more of them as well. Again, just like we talked about in males. However, one of the big differences is when does this mitosis occur?
During fetal development or gestation.
Mm-hmm. Exactly. This all occurs in utero. When you are developing inside of mommy's belly, being the overachiever that women are, they're already working on the grandkids. And in fact, at birth, there are no oogonium remaining. At birth, all the females are left with are primary oocytes. About 2 million. That doesn't sound like a whole heck of a lot. Heck, I've probably made more than 2 million sperm since the beginning of lecture. How can females get away with producing such a tiny number of eggs? Didn't we talk about the increased number was important for the genetic variation? Silence. So apparently we're not viable as a species because females don't produce enough gametes.
You only need one a month.
Exactly. There you go. Exactly. When you think about, again, as we've talked about, in a single male ejaculate, they release about 500 million. If we think in terms of, and again, this is a side note, how many eggs are released by a female during her life? You just said one a month. For how many months? 12 a year.
12 a year.
How many years?
45-ish, 50?
50? I like 50. 50 is a nice round number because that makes the math a lot easier, right? So that would be 13 to 63, which is a pretty darn huge range, but let's go ahead and go with it. So even if it was for 50 years, how many is that?
2,500.
So 12 a year times 50 years is 50 times 12 is how much?
600.
600. So when you think of the fact of how few they start with, but how few much more they even use. And again, that 50 years is a really long range to be sexually viable. I think that you can see that there's still a tremendous amount of genetic variability. So we're still reaching that point, even though these numbers are shockingly different. All right. So before birth, right? All of this is occurring before birth. Now, we know we have these primary oocytes, like we have primary spermatocytes, and they are preparing for meiosis. And what happens with these primary oocytes is that they start meiosis, but they don't get very far. They only get to prophase 1, at which point they then freeze. And they stay basically frozen at this stage until when? Puberty, there you go. Excellent. My wife just brought me something to drink and I have no idea what it is. It tastes very funny. She may be trying to kill me. If I die, at least we have the recording of this as proof. All right, excellent. Now, as protected as these cells are with their special coating that we'll talk about as well, do you think all 2 million of them last the 13 years till we get to puberty? No, by puberty, we could be down to as few as 400,000, which again, sounds like a minuscule number, but when we just finished talking about the fact that a female only is going to use 6 at the most in her life, then it's going to be okay. So what happens at puberty is once puberty starts, or once they start the female cycle, these primary oocytes are going to complete that meiosis 1 that they started, dividing into, of course, 2 cells. But here's another dramatic difference between males and females. Here in this case, this primary oocyte is going to divide unevenly into 2 cells. One cell pretty much just gets, of course, again, for these cells, we know they're getting half of the chromosomes that are replicated. Oh, don't want that capitalized. They get the chromosomes and only the chromosomes. That one cell that only gets half of the genetic material, is that going to make a good gamete? No. Remember, as we talked about, the sperm is a genetic missile. All it is providing is the chromosomes. So all of the cytoplasm, the Golgi apparatus, the rough endoplasmic reticulum, the centrioles, all of those things need to come from mom. So we don't necessarily want to split these evenly. So instead, we get this one cell that just gets the genetic material. And anybody know what we call that?
A dud.
It is, although we have a nicer name for that. It is the first polar body. The second cell also is haploid, gets half of the genetic material. But the second cell and all of the cytoplasm. And this we call our secondary oocyte. So whereas in boys, we get 2 secondary spermatocytes, in females, we get 1 secondary oocyte and 1 polar body.
It almost feels like it's cheating with the division.
What do you mean by cheating?
Well, because when there's the gamete production for spermatogenesis, it divides into 2 cells and they both have genetic material. But it seems like in this process, it's only making 1 viable cell. And so it's only doing the split process because that's all the cell knows how to do is divide in order to.
Okay, I can see where you're going with this. But remember, this stage is vitally important because remember with our gametes, we need both of them to be haploid. We need, if we're going to produce a viable offspring, we have to have 23 chromosomes from mom and 23 chromosomes from dad. And so in this case, this division has to occur because otherwise we are not going to get the 23 chromosomes necessary for the production of the gamete. But you're right, it is an uneven division because it needs more resources. And it's okay for the males to divide separately and separate their cytoplasm in half, because remember, most of that cytoplasm is going to be shed anyway. So it's all waste material. Here, it's not waste material. We need to produce a gamete that is going to be capable of dividing the trillions of times necessary to produce an offspring. So we need to make sure that that egg doesn't just have the right number of chromosomes, but it has all of the cytoplasm necessary for it to be a functional cell. And so that's why we're getting this uneven division. Now, once again, this secondary oocyte, like the secondary spermatocytes, is going to start meiosis II, but once again, it doesn't complete it. It only gets a little further this time. It only gets to metaphase II, and then it freezes at that stage. And it is here, as an incomplete gamete that has not finished the process of division, that the cell is ovulated. So notice when Ovaltine takes place, the gamete isn't even functional yet. And once ovulated, it lasts for about 24 hours. If it is not fertilized during that period of time, then that secondary oocyte basically degenerates, breaks apart, is reabsorbed by the uterine tube, and never actually completes the process. Here, completion of meiosis II is a dependent process. It only can finish meiosis II if it is fertilized by a spermatogonium. Not a spermatogonium. A spermatozoon. So if it is fertilized, let's make it green. If that spermatozoon is able to get in there and fertilize it, then and only then does it complete meiosis. Of course, if it completes meiosis II, how many cells is it going to form? Come on. How many cells are we going to get as a result of this one dividing? 2. Well, this cell
4.
divided in 2. 4 total, possibly, but yes, from this one dividing, we're going to get 2. And again, it is going to divide unevenly. One cell, the one that happens to have the sperm in it, when it completes, gets half the chromosomes. And again, all the cytoplasm. And this one we call the ovum. Whereas the other cell, nope. And of course, our sperm's got an N in there. The second cell just gets half the chromosomes. And this one we call the second polar body. Now notice, you guys, I know some of you had some questions about how many cells are formed, because you are correct. This first polar body, does it have the ability to divide as well? Yes. Does it always happen? No, not necessarily, but it certainly can. And if it does, then lo and behold, we get our 4 cells, just like we did in spermatogenesis. But notice at the end of this process, while we have 4 cells, we only have 1 viable gamete and 3 polar bodies. Whereas clearly at the end of this process in males, we have 4 viable gametes. So at its core, it is the exact same process. Mitosis, meiosis 1, meiosis 2. 1 cell becomes 4 cells. We start from a stem cell. We get to 4 cells as a result of that. Obviously, females don't undergo spermiogenesis. They end with cell-shaped cells, but their cells are very different from each other. We have 1 viable gamete and 3 cells that basically are just placeholders holding the genetic material, so that we end up with the 1 correct cell with the correct number of chromosomes in it, so that when those chromosomes come together with dad's chromosomes, we get that fertilized cell with 46 chromosomes on it. And what do we call that fertilized cell?
Zygote.
Yeah. We get that zygote. Excellent. All right. Done it here. Oh no, wrong button. Save, not erase. Done it here on the board. Let's look at the pretty pictures from the textbook and go through this again.
So what happens to the polar bodies?
What do you mean, what happens to them?
Do they do anything after that? I mean, they can't become a zygote. So what happens to them?
So great question. I love that question. And let's remember one last thing that you may or may not remember. We have looked at the anatomy of the follicle before. If you remember, it had those granulosa cells that we talked about that form the big, huge follicle. We talked about how we have the oocyte that is maturing inside of here. And if you guys remember, we talked about how there is a candy-coated shell on the outer surface protecting that egg. Does anybody remember that? Anybody remember what that's called? It's called the zona pellucidum. And again, we talked about it a long time ago. It certainly wasn't the emphasis of the endocrine system, so I don't have a problem with it. But this is a big, huge candy-coated shell. So when this one cell is dividing to produce 4 cells, guess what? That's all occurring here inside of this candy-coated shell. So at the end of the process, and I don't know if I'm going to be able to grab that. Does that work? No. All right, let's just cheat. At the end of this process, what ends up happening is those 3 polar bodies and that mature ovum are all going to be here inside of that zona pellucidum. So they're right there next to it, inside that zona pellucidum. And while you are absolutely correct, they are not capable of becoming viable gametes on their own when fertilized. But as we also know, this fertilized ovum that becomes a zygote needs to divide how many times? A gajillion to make a person, right? That one zygote's got to divide a gajillion times to become a person. And might that require the rapid mitotic divisions? Might it be useful to have a source of extra nucleic acids just hanging around in the area that could be broken down and used to make more DNA to divide a cell? Yeah, absolutely. While the genetic material isn't going to be used for anything, they're important resources. And those nucleic acids in there can be digested, broken down, and those nucleic acids can be used during the replication process. So it's worthwhile to keep it around, right? Recycle, reduce, reuse, recycle. And that's what we're going to do here inside of that zona pellucidum. So while you are right, they don't have anything to do with making the baby, they're going to be a useful resource that can be used in the development of it. Great question. Any others? So again, oogenesis involves the development of the oocyte and the follicle. This process begins prior to birth, freezes until puberty, and then continues with the process known as the ovarian cycle. And then, as we also talked about, ends at menopause. Here, we see that in utero, again, is when we have our mitosis. Our oogonium is dividing to produce our primary oocytes, producing a maximum of around 2 million. And remember, at birth, all the stem cells are gone. So once a female is born, she is born with all the gametes she's ever going to have in her life. Those primary oocytes, remember, begin the process of meiosis 1, but they arrest in prophase, and again, we should probably emphasize it, prophase 1. Obviously, if we're in meiosis 1, it's got to be prophase 1, but it doesn't hurt to write it here to remind us. And they stay suspended like that until puberty, at which point we lose close to, what is that, 80% or something like that of them, until we're down to about 400,000 at the time that the female reaches puberty. But again, in a very, very fertile life, that female is only going to Ovaltine about 600 of them. So again, far, far more than she is ever going to need. And again, not only does that involve 50 years of fertility, but it also means never actually getting pregnant. Because if you think about it, for the 10 months that you're pregnant, you're not producing more eggs in there as well. So again, these numbers are incredibly excessive and far more than she typically could need. So again, when we think about that importance of the genetic variability, it absolutely is there in females. Now at puberty, of course, as we know, it is the follicle stimulating hormone that stimulates the follicular cells, stimulates that primary oocyte to begin to grow, to begin to develop, and to cause the completion of meiosis I. Now, at puberty, many actually start the process. So we'll talk about this when we get to the ovarian cycle in just a minute. But typically, each cycle, only one reaches that stage where it matures and is released via ovulation. Of course, it could be possible that 2 or more could be released. And if that happens, what's the potential there?
Twins.
Fraternal twins.
Yep, exactly. Fraternal twins or dizygotic twins, where you get 2 eggs that get fertilized by 2 separate sperm, and you get 2 separate individuals, right? And they are genetically similar, just like other siblings are, but they're not genetically identical. In fact, you could get, that way you get a Brandon and a Brenda. And again, not to disparage, but technically it is possible if 2 eggs are released that you could actually have half siblings. How might something like that occur?
She has 2 partners in the same ovulation cycle.
Yeah, exactly.
24 hours.
So in close proximity to each other around ovulation, where one egg is fertilized by one male sperm and the other egg would be fertilized by a second male sperm. So it's actually possible to give birth to twins that are actually half siblings. All right. Excellent. All right. Now, as we talked about, this completion of meiosis is similar to males, except we get that uneven division. One cell gets all of the genetic material, all of the cytoplasm, all of the organelles. Sorry, not all the genetic material. All of the organelles, all of the cytoplasm, all those materials, and half of the organelles. That is our secondary oocyte. And we have the formation of our first polar body, which just gets the chromosomes. The second way this process is different is like in the males. Once we have our secondary oocyte, it starts meiosis II, but once again, it freezes in that process, not completing it. And so it is as that non-functional, immature secondary oocyte frozen in metaphase II that it is released from the ovary via ovulation. And it can only complete that division, complete meiosis II, if it is fertilized. If it is fertilized, it will complete the division with yet a second uneven division. Again, a second polar body or secondary polar body. And again, the first one, notice, has the ability to divide meiotically as well. May or may not. Do we really care? No. I guess we care in that we get either 3 cells or 4 cells, but we don't. But then the other one is that finally mature ovum. Great question. So with things like trisomy X and things along those lines, typically where you run into the problems is in problems of division. So, wow, that's huge. As we know, and primarily in anaphase 1. As we know in anaphase 1, that is where we have the homologous chromosomes that are separated. So one homologous chromosome goes to one side, one homologous chromosome goes to the other side, so that when the cell divides in half, you have 23 replicated chromosomes on one side, 23 replicated chromosomes on the other side. However, if that synapsin doesn't separate, what can happen, oops, that's not what I wanted. I wanted this. If that synapsin doesn't separate properly, then when this divides, what happens is both copies go to just one cell. So what ends up happening is after this first division, you would have one cell that would actually have 24 chromosomes in it, and the other one would have 22 chromosomes in it. So at the end of this process, you would have two cells here with 22. And again, if we think of them in terms of this of the sperm, right, then those would have one less chromosome than was necessary. And if this one's the one that divided, you would have, 24 in both of these. You would have an extra chromosome. And so when you get an extra chromosome, it's because of that failure of division during anaphase 1. Now, as it turns out, many of these types of divisional errors cause enough significant problems that the baby won't be able to be brought till term. So typically they will be spontaneously aborted prior to that just because they're not able to develop properly. But some chromosomes, like the X chromosome, is one that if this occurs, again, there are typically genetic problems that occur with that individual, but typically those babies can be brought to term, but they can have lots of problems associated with them, yes. All right. So, oogenesis. Questions on that? Excellent. If you guys don't have any questions, then of course that tells me that you understand this process. And if you understand this process, then as I know, being the sophisticated instructor that I am, if I have two similar processes, how many possible essay questions could there be? 3. So obviously one possible essay question that you will almost certainly see in the test bank is describe spermatogenesis. One would be describe oogenesis. And what would the third possible essay question be?
Compare them.
Right, compare and contrast them. Tell me ways that they're similar and different. So let's do that. Again, remember, you are not required to set it up this way. I just find it useful for me because I like to make sure I'm comparing apples and apples. So of course, I'm going to compare spermatogenesis to oogenesis. But to make sure I'm comparing apples to apples and oranges to oranges, I will over here have a third column with my characteristics. So someone give me an example of a characteristic where oogenesis and spermatogenesis are different.
Number of gametes they produce.
Excellent. And really, if you think about it, there are two ways. Let's make this smaller so it all fits. When we talk about number of gametes, we really can do it two ways. From one primary cell, and we could also think in total in life. All right. So from one primary cell in spermatogenesis, how many viable gametes do we get? 4, there you go. Whereas from one primary spell in oogenesis, excellent, 1 viable gamete. Excellent. And of course, during the course of a life, as we mentioned, how many gametes does a female produce, release? I mean, there's a couple ways we can think of it. How many total is she ever going to have at the max point in her life? 2 million, right? 2 million at the max, right? If we think about how many are, 2 million max, we can think in terms of how many are released, right? We said around 600, right? However you want to describe that. And again, of course, for males, how many is the max that they make? Yeah, gajillion.
Infinity.
Yeah, infinite. There you go. Infinite number. To infinity and beyond. There you go. All right. I'll never watch Toy Story the same way again. All right. Excellent. What's another way they're different? Exactly.
How long the process takes.
Excellent. How long does it take from stem cell to mature gametes in males?
60-ish days, was it?
60 days to get sperm, 60 to 70 days to get sperm-shaped cells. But then remember, we need an additional 20 days to mature. So around 90 days. And what about for females?
13 to 50 years.
Yeah, hopefully it's not 13. Hopefully it's a lot more than 13. But you're right, 13 would be the earliest that it could occur, right? Of course, we're hoping most 13-year-olds aren't getting pregnant. As someone with a 13-year-old and a 16-year-old, that's definitely not what we're rooting for. But yes, absolutely, it is a much, much longer process, right? So 13 plus years, depending on when that can be. Excellent. What else? Give me another way they're different.
The role of meiosis, like in the male, it's kind of a linear process, and in females, it's like start stop.
Okay, excellent. Hey. I like that. Again, this is another kind of way of timing of the process. So you're absolutely, this is in males, it's a continuous process. Once it stops, it continues. Whereas in females, as you mentioned, it starts and stops really twice. And along those same terms, right, this is an independent process. Obviously, these require hormones for this to occur. But once we start the division of that stem cell, you end up with 4 gametes. Is the same thing true here in oogenesis? Is it an independent process? No, it's very much a dependent process. In females, this process doesn't get to complete if it is fertilized by the sperm. Right? Excellent. I'm sure there are a dozen more things we could think of, but again, the point is you get the idea. This is what I'm looking for. So notice we can compare the processes. We can describe each individual process. Make sure you understand how to do this. All right. Questions on that? All right, excellent. So let's go back to this slide. Because as I mentioned with the female reproductive system, nothing can be quite this simple. We just talked about how this compares to sperm development. But also, as we talked about, the female gametes aren't maturing by themselves. As we talked about, they have those support cells, that follicle that is surrounding it, that is also maturing and developing and changing in this process as well. So while we can absolutely positively talk about oogenesis, we also need to be able to talk about well, let's start here. of the follicle cells that support cells for it. And that process is actually what we call the ovarian cycle. And so now we have to relate the events that we know that's going on with the oocyte with what's going on with those follicular cells inside of the ovary as well. Now, your book has this awesome and incredibly terrifying looking table that does a good job of kind of relating all of what's going on in the hormones to all of what's going on to the ovaries, to all of what's going on in the uterus as well. And so we have to make sense of all of this. And so that is what we're going to do. Now that we know what's going on in the egg itself, we are able to talk about the ovarian cycle. And so that is what we're going to do next. However, like before, I want to come at this fresh. So let's go ahead and take our second break here. So it is 10:27. So we will return back in 15 minutes. That'll be what, 10:43? And I will start the recording at this time. So at this point, we have talked about what's happening. Again, notice everything we've talked about so far is just what's going on in this, that's a horrible color, green. This little cell here in the center of this structure that is ovulated. But obviously, as you can see, there's a whole lot more going on with those follicular cells. So now we have to relate the two things together. All right, and we will do that after the break. I will see you guys in 15 minutes. All right, excellent. Now, I really like this illustration from your textbook. We're getting ready to talk about the ovarian cycle. The only problem that I know people have sometimes had in the past, so I want to make sure I emphasize it right here so that we don't have this issue, is that the point of this illustration is to basically show all the stages. Notice it shows basically 8 stages in this process, and they wanted to put all of them here. However, it's important to know that these follicles do not migrate around in the ovary. So basically what happens is that all of these stages that are shown here might happen with this follicle right here during this ovarian cycle, and they will all happen right at this location. Then the next time it'll happen right here with this cell, and they'll all happen in this location. And then the next time it may be in the other ovary that it takes place. So these follicles don't move around. So again, I've had a couple people in the past be confused by that. So I want to make sure I emphasize this. This just shows all of the stages. They don't move, but they all just occur at that one location where you'll see all these things occurring. All right. So let's work our way through this. Basically, development of the ovarian follicle occurs in 2 phases. They are the follicular phase, which is basically day 1 to day 14, and the luteal phase from day 14 to day 28. Notice, as we talked about, this gives us a 28-day cycle, which is a good, decent average for this. However, does everybody truly have a 28-day cycle? No, some may be shorter, closer to 25. Some may be longer, closer to 31, something along those lines. So there is going to be variability from person to person. I will tell you right now that that variability typically is more in the first phase. Now, the other side of this is once a female has an established cycle, is she likely to be 23 days, 1 month, and then 31 the next? No. So while different women will have different cycles, typically once it's established, whatever their cycle is will be fairly consistent. So again, where there's the changes primarily in the follicular phase as opposed to the luteal phase. And of course, it should be obvious, but what do you think the big dividing process or stage or event that occurs that separates the follicular phase from the luteal phase?
Fertilization?
Not necessarily fertilization, because that's not necessarily hopefully happening every month, but what would the event be that would lead to fertilization?
Ovulation.
Ovulation, there you go. Now you're right. Once ovulation occurs, then absolutely fertilization can occur after that. But like I said, hopefully a female isn't fertilizing every month. There's that ovulation in between. So that's typically the event that separates them. Your book divides it into 8 stages. And as you know, I don't care so much about the numbers as long as you understand all of the steps in it. So let's do this. Let's actually do it first in the drawing. Then we'll look at all the pretty pictures from all of the textbooks and make sure that we understand this. So let's go ahead and clear that. We are going to do the ovarian cycle. And as we know, the ovarian cycle starts at puberty. We'll talk about the hormonal events that trigger this in just a minute. But remember, at puberty, basically all of the oocytes are primary oocytes. And again, they're frozen in prophase 1. And they are all going to be surrounded. So let's go ahead and make that cell. Here is our cell. Make it big enough so that I can write in there, even if it's tiny. It is going to be surrounded by a single layer of flat cells. So a single layer of squamosal cells. And this single layer of squamosal cells are called follicle cells. This structure, the primary oocyte frozen in prophase 1, surrounded by a single layer of follicle cells, is what is known as a primordial follicle. So we have this primordial follicle, which is that primary oocyte frozen in prophase 1, surrounded by a single layer of flat cells. Now, many of these primordial follicles are going to start to mature. But this is a very slow process. It can take up to 1 year for this maturation to occur. So many of these, I need to make that smaller, are going to start this maturation process. But only a dozen or so, dozen plus, reach the next stage eventually. So in time, in time, slowly, we are going to get the point where our egg is getting a little bit bigger in this process, but it is still a primary oocyte, frozen in prophase 1. So again, it is growing, but it hasn't completed meiosis 1 yet. But the big change that has taken place is in those granulosa cells. Those granulosa cells have matured from a single layer of flat cells into a single layer of cuboidal cells. These cuboidal cells are what are known as granulosa cells. And as we talked about, it is these granulosa cells that produce the estrogens. Now, of course, to produce those estrogens, we need the thecal cells. So we're going to start to get the formation of thecal cells on the outer surface. Oops, I don't know where I'm writing that. And we can use blue for that. And this structure, the primary oocyte with a single layer of granulosa cells, is what is known as a primary follicle. So notice we went from a primordial follicle to a primary follicle. And like we talked about, dozens reach this stage. Each month. Now, someone remind me again, what hormone is responsible for driving this process?
Luteinizing hormone?
Is it luteinizing hormone?
No, that would make sense. Follicle stimulating hormone.
Excellent. This process is driven by follicle stimulating hormone. Follicle stimulating hormone is starting the maturation of those primordial follicles into primary follicles. But then something really interesting starts to happen. Oops, that is off the board. There is a drop Oops. There's a drop of follicle stimulating hormone. And at this point, the race is on. Of those dozens or so primary follicles that have started their division, they are going to continue to grow and continue to expand, but they're now going to be competing for follicle stimulating hormone. Again, we still have our primary oocyte, frozen in prophase 1. I'll just write it that way so I don't have to keep writing it out over and over again. But now, it is surrounded by 2 or more layers of granulosa cells. And a more intact thecal cell layer on the outer surface. So again, we have thecal cells surrounding two or more layers of granulosa cells. And this is what we call a secondary follicle. And only about a handful. Nope. Again, we're not talking about the oocyte. We're talking about the follicle here. Absolutely. And you're right, it's not the end. And at this point, only about a handful remain. These follicles require the follicle stimulating hormone. And as there's less and less of it available, they have to compete more and more for that's viable. And so those that are able to survive with less follicle stimulating hormone, or those that get more follicle stimulating hormone, are going to be more successful and are going to be able to survive and are going to be able to grow. And the ones that don't are going to be broken down and reabsorbed and basically go away. Now, one more interesting thing happens during this second follicular phase. As our follicle grows, maybe too big and small. There we go. Let's be more specific. There is a production of many more granulosa cells. And so what starts to happen is our follicle starts to expand beyond just surrounding the oocyte. And so what will start to happen, and I'll just cheat and grow it on this one, draw it on this one here. What will start to happen is that some spaces will start to form. That's not going to work. Do it this way. Some spaces will start to form between the layers of cells. Nah, that looks horrible too. All right, never mind. I'll just draw it. There's our oocyte. Here's our first layer of granulosa cells. Second layer of granulosa cells. But what starts to happen is there starts to be a separation between the layers of the cells. And these separations basically fill with interstitial fluid. So basically what happens is we start to get the formation of these fluid-filled spaces between the layers. Now, these are still considered secondary follicles at this stage, but I make a point of emphasizing this because as the cells grow more and more and more, they begin to dramatically outpace the formation of the, or the growth of the oocyte. So while the oocyte gets nice and big, what ends up happening, and now I'm just going to draw it with a highlighter now, we get the layers of cells, here, let's do it this way. We get the layers of cells that surround it, but then what happens is we get this formation of this big fluid-filled space. And this big fluid-filled space we call the antrum. So these small individual pockets expand to perform a structure that is much, much greater than the cell itself. My scale is slightly off on this. Oh, and that's the other thing I forgot to add to all of this. Let's go back a step.
You said the antrum is the enlargement of these fluid-filled spaces?
Correct. The fluid-filled spaces enlarge and expand and fuse together. So it becomes one singular large fluid-filled space that we call the antrum. Remember also, around this primary oocyte is that zona pellucidum, that candy-coated shell of glycogen-rich protective layer. And let's put it here as well.
Is that not a part of the primary follicle or the primordial follicle?
It will start to form here, but as it's starting to form, it may not be complete and it may not be as easy to see. It's not here in the primary, but as it starts to, it's like the thecal cells. The thecal cells are starting to form in the primary. The zona pellucida is starting to form, but typically they're not formed enough where you fully see them. By the time they get to the secondary and tertiary follicle stage, and that's where we're at here. Here we have a tertiary follicle. Or what is also called a mature follicle, or what is also known as a Graafian follicle. All of those are acceptable terms for that. We have this big, large fluid-filled space. Our oocyte has finally completes meiosis 1 and starts meiosis 2. But does it get a chance to actually complete the process?
It's not.
No. So this cell inside of our mature follicle is a secondary oocyte frozen in metaphase 2. Now, we have already identified the zona pellucidum. Yes. But now we are going to give a special name to these granulosa cells. Now, technically, and let me draw a box. This granulosa cell right here and this granulosa cell right here, is there anything different, do you think, between these two granulosa cells?
Only location?
Exactly. The only difference is location. Their functions are exactly the same. They're producing the estrogens. They're helping to support and mature this cell, but one is in direct contact with the egg. And the ones that are in direct contact with the egg basically form a crown around its surface. And if we've learned anything in the past year and a half, what's the term that refers to crown?
Corona.
There you go. Forms a layer we call the corona radiata. Again, there's nothing special about these cells other than their location. And typically in a single cycle, one cell and one cell only, or one follicle, I should say, and one follicle only reaches this stage. And all of this to get from this point. Now, again, remember, this is taking months for these primordial oocytes to occur. But as we've talked about day 14 of the cycle is when we have this mature graphene follicle, at which point there is a huge spike of luteinizing hormone. And when there's that huge spike of luteinizing hormone, what happens?
Ovulation.
Ovulation. Ovulation occurs. When ovulation occurs, basically the secondary oocyte frozen in metaphase 1 and the corona radiata that surrounds it, these cells are released. This basically big balloon bursts, releasing that secondary oocyte and the corona radiata that surrounds it. But notice there's a massive number of follicular cells that remain. And that's the second function of that luteinizing hormone. Luteinizing hormone transforms, it transforms the remaining granulosa cells. And it converts them into this big, huge glandular structure that we call the corpus luteum. So basically, at first, this thing looks like a big deflated balloon, because that's what's happened. It's ruptured, and it's become this big deflated balloon as that's occurred. But then this structure glandularizes and becomes this big, huge, massive glandular structure, which, as we know, produces both estrogens and progestins. Because remember, progestins are that chemical signal that tells the body, beware, the egg has been released. Fertilization can take place any second now. Get ready. But what happens is this corpus luteum starts to degenerate, starts to break down. And ultimately, as it degenerates and breaks down because of a drop in luteinizing hormone, so there's a drop of luteinizing hormone, leads to the degeneration of the corpus luteum until basically it shrivels up into scar tissue. That we call the corpus albicans. Albicans, again, albino, whitish coloration. It's just basically this non-glandular tissue, no longer functional, at which point the cycle begins anew.
So the secondary oocyte that was ovulated, does it become an ovum now?
If it gets fertilized. But remember, if it doesn't get fertilized, then again, it's viable for about 24 hours after it's released. At which point, if it's not fertilized, it degenerates, right? And again, when it's ovulated, it is released into the uterine tube. And unlike the sperm, it can't propel itself. So it basically, hopefully, gets into the uterine tube. It doesn't always. We'll talk about that in a bit. But the goal is to get it into the uterine tube. And if it's fertilized, that fertilization will take place in the uterine tube. But it's only viable for about 24 hours. So if it's not fertilized during that time, then it's going to basically degenerate, be broken down, and go away.
So each month you only have 24 hours to get pregnant, technically. Then.
Well, the egg is viable. I don't think we talked about that. The egg on release from the ovum is viable. I mean, from the ovary is viable for about 24 hours. Sperm, when ejaculated, are viable. Most sperm are viable for about 24 to 48 hours. Although there are some sperm, and this is not my term for them, there are some what they call super sperm, which can be viable for as long as 72 hours. So if you think about it, there's basically about a 3-day period of time where copulation and ovulation have to occur for fertilization to take place.
Question.
Yes.
So in this process, if something goes wrong, is that how an ovarian cyst is formed?
No, ovarian cyst. So an ovarian cyst typically doesn't have much to do with the follicles themselves. It can be related to it, but remember we finished talking about how the bulk of the structure of the ovary is this fibrous tissue, that tunica albicans. And because it's a fibrous tissue, what happens is in some areas, that fiber can come a little bit more dense and form a little bit of a pocket, and that pocket fills with fluid. And that's basically what a cyst is a fluid-filled fibrous pocket. Again, often they rupture, they form, they rupture, they come and go, and the female's typically not too aware of it. If it becomes large enough, in some instances, it can become somewhat painful. But most of them are small enough where they don't cause any kind of major issues that way. And they don't typically have a significant effect on ovarian function either. All right, I've done a poor job of drawing this. Let's look at the pretty pictures from the textbook and go through this again from this. So again, here we see an example of some primordial follicles. So again, notice at the beginning of the process, we have many of these cells that start this. And notice here we have that primary oocyte frozen in prophase 1. Here's a primary oocyte frozen in prophase 1. Here's a primary oocyte frozen in prophase 1. And notice all of these have a single layer of flat cells on their outer surface, those follicular cells. And so this entire structure, this primary oocyte frozen in prophase 1, surrounded by a single layer of flat cells, is what we call a primordial follicle. However, notice on this slide, there's one other very interesting thing. We have a primary oocyte frozen in prophase 1, but notice it's a little bigger than the ones next to it. And more importantly, instead of a single layer of flat cells, notice this one has a single layer of cuboidal cells on its outer surface. So while these first three things that we drew were primordial follicles, what is this second thing that I just drew?
Primary follicle.
Exactly. So notice in a process that can take up to a year, these primordial follicles will mature into a primary follicle, where those single layer flat cells become a single layer of cuboidal cells. And absolutely, that is a great example of a primary follicle. Now notice here, a little bit more mature. Notice our oocyte has gotten bigger, but identify this cell, be very specific.
Primary oocyte.
And you'd get partial credit for that on the exam.
Frozen in phase 1.
There you go. Primary oocyte, frozen in prophase 1. I guarantee you that question is in the lab exam bank of questions, and it will have those magic words, be specific, which tells you I wanted you to be as precise as you possibly can be. So this is a primary oocyte frozen in prophase 1. Notice you can see the tiniest bit of that zona granulosa starting to form, forming kind of a distinct boundary here between the oocyte and the granulosa cells. We can see a little bit of the thecal cells starting to form, condensed down on the outer surface. But again, they're not super distinct. What is super distinct is that single layer of cuboidal cells, those granulosa cells that wrap around it. So again, it can take up to a year for these primordial to reach this primary stage. Each month, dozens reach this stage. So each month, we get dozens.
Why is it more correct to say that this is a primary oocyte frozen in prophase 1 versus saying this is a primary follicle?
Great question. The thing here in pink is a single egg, a single cell. So this thing that I've highlighted in pink, let's draw the whole thing. This whole thing here in pink is a single cell. This is one cell. On the outside, this is also a cell, and this is a cell, and that's a cell, and 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 cell. So the thing in the center is the egg. That is the oocyte that is developing. This egg with the support cells that surround it are a primary follicle. Remember, follicles are formed by the egg, the developing egg, and the support cells around it. So a follicle is not just one cell. A follicle is a structure made up of many cells, whereas the egg is just one cell. So that is why this is a primary oocyte that happens to be in a primary follicle. Whereas if we go back to the previous page, we have primary oocytes in primordial follicles. All right. So a follicle is the egg and the support cells that surround it. It's the whole structure together. Whereas the oocyte, at whatever stage it is just that one cell. Does that make sense? Okay, excellent. So again, we get the formation of those granulosa cells, our zona pellucidum starting to form, our thecal cells are starting to form. Our thecal cells are producing androgens, and those granulosa cells are converting it to androgens. And so here at the primordial phase, we start to get the production of estrogens. And those estrogens, among other things, lead to the growth and formation of our egg. Now, our granulosa cells continue to divide, continue to expand until they form a secondary follicle. Again, a secondary follicle is comprised of an egg. And what is the stage of the egg? So again, on this picture was on the exam, and I had an arrow like this, and I asked you to identify this cell. How would you identify this cell?
A primary oocyte.
And you get partial credit for that on the exam, because I want you to be specific. So it's a primary oocyte. Finish it off. Anyone?
I can. I like the requisite knowledge.
Frozen in metaphase 1.
Prophase one.
Prophase.
There you go. Excellent. Plus 2 or more layers of granulosa cells. So again, a secondary follicle is both of these things. The primary oocyte frozen in prophase 1, and in this case, 2 or more layers of granulosa cells on the outer surface. So we see those multiple layers of granulosa cells. Again, notice our zona pellucidum is getting a little bit more distinct. Thecal cells are starting to get a little more distinct on the outer surface. Start to see those cells forming out here on the outer surface. And like I said, at this point, we have a handful that get to this secondary follicle stage. Now, like I said, the problem that happens is we start to get a drop in the levels of follicle stimulating hormone. We'll talk about why in a bit. So at this point, it is a true race, right? Can you get enough follicle stimulating hormone? Can small amounts be enough to sustain you and allow you to grow? And if you don't get enough, or if you grow well enough with small levels, then those follicles degenerate and die. And only one is going to reach that tertiary stage. But those that are growing, and I think I have to erase this because I have to show it here. Notice as these follicles continue to expand, and now here, really, really pretty. You can start to see the distinctions. Very distinct thecal layer on the outer surface here. And here, as we look at the granulosa cells inside, I don't remember what I used for the granulosa cells. Was it green? But the point is, you can start to see some separation occurring. These fluid-filled spaces are starting to occur as we're getting the separation of these cells, as the follicle is expanding much more rapidly than this cell inside. So even as these continue to expand, it isn't until all the fluid-filled spaces fuse together that we have one single massive fluid-filled space that is typically bigger than the egg itself, that we have our tertiary follicle, or the Graafian follicle, or that mature follicle. All are appropriate names for it. Notice here, we have some of the anatomy we've already talked about. We know we have these thecal cells on the outer surface. We know we have our granulosa cells forming the big, huge follicle. But remember, we also mentioned how some of these granulosa cells are surrounding the egg. And what do we call that layer of granulosa cells that surrounds the egg?
Corona radiata.
Corona radiata. Notice also we can very, very clearly now see that glycogen-rich clear acellular layer that is the zona pellucidum around the egg. We, of course, have our fluid-filled antrum. But there's been one other big change that has taken place. Now, when I have this picture on the exam, and I ask you to identify this cell and to be specific, I want to make sure I'm not pointing just to the nucleus. When I ask you to identify this cell and be specific, what is your answer to this going to be?
Secondary oocyte frozen in metaphase 2.
There you go. Beautiful. And of the dozens that started the race to begin with, Typically, how many follicles reach this stage?
1.
Exactly. Only one follicle reaches this stage each month. Again, this typically happens by day 14, but as we said, it can be shorter than that. As early as day 10, as late as day 17. This first phase is where there's the most variability. That large fluid-filled antrum, that corona radiata on the outer surface, and our oocyte has completed meiosis 1 and starts meiosis 2, but freezes in metaphase 2. Here, we see this nice big fluid-filled antrum, but this structure expands dramatically. Here, we actually see that follicle bulging out. So notice if you look carefully at this picture, you can see, that's horrible. Let's go with a darker color, bigger color. This is that expanded follicle, like a big bubble. Here, of course, is the egg with its corona radiata around the outer surface. Yeah, exactly. And when there's that huge spike in luteinizing hormone, this ruptures. And that egg is released into the abdominal pelvic cavity. Now, in most cases, it is encouraged into the uterine tubes where it can then be fertilized. But does it have to enter into the uterine tube? That's not how you spell encouraged. Does it have to get into the uterine tube? No. Of course, if it doesn't get in the uterine tube, then it's likely won't be fertilized. And in the rare instance that it is fertilized, it won't be able to find a place to make purchase. And on the rare instances where it does find purchase, is it going to be able to be brought to term and be a viable baby? No. What do we call that condition?
Ectopic?
Yeah, an ectopic pregnancy, exactly. An ectopic pregnancy is where that fertilized egg is able to miraculously find some other location other than the uterus where it is able to attach and start the growth process, but it absolutely positively is not something that can be brought to term. So it is something that isn't going to be viable. But the goal is to get it into the uterine tube and get it fertilized. So again, that secondary oocyte with its corona radiata is expelled into the pelvic cavity. The egg continues on its journey and is no longer a part of the ovarian cycle, but remember we have that huge follicular cells that remain behind. Of course, these follicular cells that remain behind are transformed by our luteinizing hormone, which is why we start the luteal phase, and become a structure known as the corpus luteum. Here we see an example of a corpus luteum. Notice again, as I talked about, it kind of looks like this deflated balloon. And that actually reminds me, I wanted to go back to that one picture at the very beginning. Notice this structure right here is that deflated follicle after rupturing occurs. Notice it starts as something like this. Obviously, this plane of section doesn't have the egg in it, but we have this big, huge fluid-filled antrum that when it bursts and releases, basically looks like a collapsed, deflated balloon. This is the beginning of the formation of our corpus luteum. Notice this ovulation is a fairly violent act. If you happen to just run across an ovary that is laying on the ground, you can actually pick it up and look at its outer surface. And by counting the scars on the outer surface, you can get an indication of how many times it is ovulated an egg, which could give you an indication of the relative age of the individual who dropped their ovary on the ground. Now, does that mean that women are able to perceive this event when it occurs? I'll say some people shaking their head no, and some people shaking your head yes, and you're both correct. Not every woman is, but there are some women who are actually able to perceive it. It's a condition, they have a German name for it called Mittelspurz, which is basically they get this twinge or this pinching sensation in their side. And again, in most cases, they don't know why it happened. Maybe they just twisted or turned wrong or something like that. They're not aware of it. When I was a young instructor, I taught for a summer at Woodland Community College. And the department chair there was trying to get pregnant at the time. And it wasn't until she was trying to get pregnant and actually actively monitoring her cycle that she realized that she was one of those people who was able to experience it. You know, it's one of those things every once in a while you feel a little twinge in your side. You don't think twice about it. It wasn't until she was following her cycle more closely that she realized that she was actually able to perceive that sensation. So not every woman's able to, but there are some who can. And so, like I said, it forms this, what first appears to be a very deflated balloon type looking structure. However, as we mentioned, that luteinizing hormone is going to cause a massive proliferation of these cells, a massive growth and formation into a huge, massive glandular structure. Here, I was going to do the histology later, but this is a good time as any to do it. We'll get back to this in a second. There we go. So you can go from that collapse shaped structure. See, there you go. I like that picture so much I grabbed it. To where it can form this big, huge, massive glandular structure that forms in the ovary. There's the corpus luteum there, or I think this next one does an even better job of showing it. Here, this big, huge, dark glandular structure is that corpus luteum that forms. A big, huge, massive glandular structure. And remember, the key to this big, huge glandular structure is, yes, it is producing estrogens, just like the granulosa cells were, but it is also producing those progestins, which, like we said, are the warning bell hormones that tell the female body to get ready. The egg has been released. Fertilization could occur anytime now. You could get pregnant. Get ready.
The slide you just showed, it was like a very smooth round. So does that mean it was like right after it got rid of the egg before?
No, the opposite. When it first gets rid of the egg, it looks like that deflated balloon. But what's going to happen is that the luteinizing hormone cause all these cells to divide and expand. And so what typically happens is it divides and expands and becomes this big, huge, singular glandular structure that forms. And so when it first is expulsed, it looks like this. And then as it grows more and more glandular cells, then it becomes typically a large, solid structure. All right. Now, this is formed by the corpus luteum. Pardon me, formed by the luteinizing hormone. But it is also maintained by the luteinizing hormone. And that's a problem because after the big spike, luteinizing hormone levels drop significantly. Oops. There we go. Drop significantly. And so when they start to drop and it's being maintained, what starts to happen is the corpus luteum starts to degenerate. It gets smaller and smaller, producing fewer and fewer hormones until ultimately it shrivels up into this. Notice the white coloration of it is because it is no longer glandular in nature. It basically shrivels up into this non-functional piece of scar tissue known as the corpus albicans. At this point, our progestin and estrogen levels decrease rapidly, and a new ovarian cycle begins. Of course, unless fertilization occurs, and then, right, all sorts of craziness happens after that, but we'll worry about that later. Let's just focus on the ovarian cycle. And I think, there we go. Here's our review cycle. So again, notice, oops, do this here first. We start the follicular phase, development of the follicle, starting from primordial, right, which are these little dots in here, to the primary. Primary becomes secondary. Our secondary keep getting larger and larger till they produce the fluid-filled spaces and then become that big, large tertiary, which then leads to ovulation. The remaining granulosa cells that stay behind are transformed into this big, huge, massive glandular structure known as the corpus luteum, which ultimately shrivels up into this tiny little piece of scar tissue known as the corpus albicans. And then the process begins again. We use the pictures from the textbook, we use the illustrations from the textbook, but as I kind of hinted at and showed you, I have these great actual pictures of the ovary that we can use to see these stages as well. Notice here, as we can see, it's a relatively low magnification view, but notice as we can see, as I mentioned, this is a cat's ovary that has been injected with a large amount of follicle stimulating hormone. So we can see the massive number of big, huge antrums from all these massive numbers of tertiary follicles. Yep, great question. Yes, the corpus albicans stays forever. So again, not only can you look at the scarring on the outer surface, but you can look at the internal scarring to get some sense of age as well. Now, notice I see lots of tertiary follicles here, but I don't see lots of eggs. Why is that?
Just how it's cut?
Exactly, right? If you think about it, if you had a balloon, right? That balloon was that big, huge filled follicle, and the stem that you fed into the balloon, the air into the balloon, is about the size of the egg. So what are the chances of you slicing it just so perfectly that you're going to get a little piece of the egg? Not so good. So there are some eggs in here that we can see. But notice also we can see other stages. Here we can see some secondary follicles. We can see it's probably, again, it's probably too low of a magnification, but these are probably close to primary and primordial, probably in that phase. But, oops, didn't mean to do that. Let's go ahead and take a closer look. Here's another cat ovary where we can see that anatomy of it very, very nicely. Excellent. Here are our primordial follicles. Notice again, as we can clearly see here, we have a cell surrounded by a single layer of flat cells. What do we call the single layer of flat cells?
Thecal cells?
Yeah, follicular cells, excellent. Single layer, follicle cells, follicular cells. Excellent. And then what do we call the egg?
Primary oocyte frozen in prophase 1.
Excellent. And all of that together, we call our primordial follicle. Now notice also, primordial follicles are not just randomly distributed throughout the ovary. Notice instead, they're clustered. That's a horrible color. They're clustered together in these structures. And any idea what you might call a cluster of eggs?
Good. Not a clutch.
Close. How about, and carton would be good too. A nest. These are egg nests. So these, so you have these egg nests where they're just these clusters of these primordial follicles together. Notice though, oh, great picture. Here, we can see one of them starting to mature. Notice we're starting to see the formation of those cuboidal cells on the outer surface. Here, we're starting to see some cuboidal cells form. We can see those cells getting bigger, the nuclei getting bigger. We are starting to get the formation of some primary. So again, here we see plenty of primordials, but again, notice here, we see the beginning of a primary follicle. And I think I have one more picture that shows it nicely. Here's another primordial. That's obviously got to clear that. Here we're seeing the formation of a primary follicle. Here we're seeing the formation of a primary follicle. So we can see these primary follicles. We can see the sizes aren't that much. The egg in the center, which is still a primary oocyte, frozen in prophase 1, isn't much bigger than the primordial ones, but it's really the granulosa cells on the outer surface that are changing. All right. Notice also, as it starts to go to the secondary phase, not only do we get more layers of granulosa cells, but now the egg itself is also getting much, much bigger. However, even though this egg is getting much, much bigger, how do we identify this egg? Identify the cell. Be specific.
The same.
Yeah, it's still a primary oocyte frozen in prophase 1. All right. Here we see an early secondary follicle. Here, notice we start to see a little later of a secondary follicle because you can start to see some of the separation, some of the space that is forming between the layers. Notice also we can see that much more distinct zona pellucidum that is forming around the egg as it's forming. Again, what stage do you think this is?
Since the antrum started to form, is that why it's now the secondary in metaphase 2?
So it's a good guess. And that's one of the reasons why I have this picture here, because I appreciate it is tricky. In this particular one, you're right. The fluid-filled space that is forming primarily in one location. However, compare this one and this one and this one to that. Notice while these fluid-filled spaces are forming, they're similar in size or even smaller than the egg. And even if it's in one location, if they're similar in size or smaller than the egg, they're still secondary. So this is still a secondary follicle. This is still a secondary follicle. This is still a secondary follicle. But here, now we see the dramatic difference in the size of that big, huge, massive fluid-filled space that is the antrum. Notice the other advantage of having this big, huge fluid-filled space that is the antrum is we can also more clearly see the corona radiata that surrounds it as well, that belt-like structure of granulosa cells that surrounds it. So here we have our first example of a tertiary follicle. And now that we have a tertiary follicle, when I ask the question for you to identify this cell, be specific, you finally get to say something different. And what's that something different that you finally get to say?
Secondary oocyte frozen in metaphase 2.
Perfect. Again, here's another tertiary follicle. Another tertiary follicle, all right, plenty of examples of that. These are then going to rupture and burst, releasing their contents, becoming this deflated balloon structure that is transforming into the corpus luteum. You would still call it the corpus luteum at this stage, because if ovulation does occur, it is transforming. This is the corpus luteum, but this is a very early corpus luteum. As we mentioned, that corpus luteum can expand to become a huge, massive glandular structure. That eventually deteriorates and goes away. Notice here, we see some examples. And again, it's usually easier to see. It's compact, it's lighter in color. Here we see a lot of examples of a relatively mature ovum, because we can see a massive numbers of these corpus albicans. As someone mentioned earlier, they do stay forever in the ovaries tissues. So basically it's these kind of convoluted, typically clear colored structures that is that corpus albicans. Questions on that? All right, excellent. We have covered a lot today, but notice if we come back to this graph, really, or this illustration, this chart, all we've covered so far is just what's going on right here. We still have to talk about how this process, although we've hinted at it, we need to be more precise about how this process is controlled by the hormones of the anterior pituitary. We know these follicular cells and these corpus luteum cells are producing estrogen and progestins. We need to talk about the effects of those on the body, on the maturation of the follicle, on the anterior pituitary, and on the uterus and the phases that are going on there. So while we have covered a lot of material today, as you can see, we still have a lot more to do as well. All right, but this is where I wanted to stop for today. It's a teeny bit early, but I think we've covered a lot of material very successfully. So any questions on this before we call it a day? Let me go back real fast before I do that. Again, I've shown you, I think, all the histology we want. We looked at the penis, we looked at that, so that was good. We've looked at the ovaries. We don't need to look at the uterus or the uterine duct yet, so we're good. Yeah, we've covered all the histology I wanted to cover today too. We snuck that in as we were talking about the other things, and we've talked about all the processes that I've wanted. Yes, thank you. Absolutely. So actually, while I'm thinking of it and while I have you here, let me go ahead and do that right now. So absolutely, I will be opening the exams for today so that you have the opportunity to take a look at them. Please do take a look at them. My office hours start at 12:30. I need to run a quick errand first. So even though we're finishing a little bit early, I won't be in my office right away. But my guess would probably about 12:20 or so. I will be in there. So if you have any questions or want to look at it, I'll make it available so you can look at it now. But if you have any questions about it or concerns, you can come and see me during my office hours and we can talk about that. So, all right, let's see here. All right, your lab exam has been released. And your lecture exam has been released. So again, these will be available for about, there you go, for about 24 hours. And so around this time tomorrow, I will be turning them off so that you guys have had your chance to look at them. So again, if you have any questions, email me or come to my office hours. Thank you for the reminder of that. I appreciate it because I would have forgotten. All right, any other questions? All right, excellent. Then you guys have an excellent day and I will see you Wednesday.